Ahora comenzaría el panel 4.2, con lo que este panel termina y ya toma la palabra.
Bueno, ya que ayer estamos en el problema porque ha sido. Vamos a intentar. Sí. Vamos a intentar resumir ahora cuáles son las propuestas. Sobre todo, por favor, os pido, ya sé que son los últimos, tanta gente ha sido difícil un poquito controlar, pero yo os pediría un esfuerzo de que quedéis clara la idea, porque ahora tenemos una sesión de networking hasta las 5 de la tarde en un hotel, en un ambiente relajado, donde ahí pueden exponérsela. Habrá una mesa con un cartel para que los interesados en torno a ese tema se puedan poner y puedan comer juntos, conversar. Ese era el diseño de la función, ¿no? Después de haber. Entonces, más una presentación breve de lo que queréis comentar. El primer ponente viene de Suecia, el doctor José Pedro Gil. José Pedro es un amigo de muchísimos años, una persona que con una trayectoria amplísima. Desde Portugal es profesor en el Instituto Karolinska de Estocolmo, que supongo no hace falta decir que da el Premio Nobel de Medicina, pero sobre todo su actividad internacional ha estado ligada a las enfermedades infecciosas, en concreto a la malaria, y se puede decir que es aquí líder en estudios de malaria y principalmente en África, ¿no? Entonces, Pedro nos puede aportar a nivel de networking, lo hablamos hoy, una visión no solo desde la península ibérica, sino desde el norte de Europa y proyectando todo su trabajo a África. José Pedro, muito obrigado. Thank you very much for your effort and to come here that long is something that we really appreciate. Thanks.
My pleasure. Hello, you're alive? I'm still alive, at least. So, as Adrian said, I come from a bit far away from Sweden, even though I work a lot also in Portugal, especially with the Tropical Institute, because, as I said, I work with malaria. It's a little bit different from until now, I think, a little bit out of Europe, and a little bit different because it's an infectious disease. And personalized medicine for infectious diseases will need to deal not only with the host, but also with the enemy, with the pathogen. So we have to deal with those two things at the same time. And that's the challenge I've been pursuing for a long, long time. So here we are with the case concept, which is malaria, which is a serious one with 600,000 deaths every year. If I speak too fast, just tell me. People sometimes complain. So for the last, say, 15, 20 years, I've been mainly basing my research on clinical trials, efficacy trials of drugs, antimalarial drugs in Africa. I started, well, in East Africa, and somehow I kept on moving west. And nowadays, I mainly work in West Africa, in Mali, in Burkina Faso, and very much with the Portuguese-speaking countries, African countries. So that's a bit the basis of our research there. We do a little bit more things, but not going to speak about that. But as I said, we have to deal, in this case, with the parasite and with the person. And we have to understand the pharmacogenetics of the both. In the human is pharmacogenetics. In the parasite, in the enemy, usually we call it drug resistance. Well, it's like an extreme case of pharmacogenetics for the animal. So I'm just going to pick up a number of, say, projects. The main ones that are running right now in my lab, in a way crystallizing the idea that we have to understand both sides of the drug, the host, and the pathogen. Well, artemether-lumefantrine is the most used antimalarial in the planet. It's about 700 million doses every year, and it's the backbone of the malaria control programs in Africa. And unfortunately, the drug that until now has been very resilient to resistance, the last years in Angola, it seems something is going wrong. We performed a clinical trial there about 2 years ago during the COVID pandemic. Go figure, it was not easy. But anyway, we discovered one thing. We discovered a marker for the resistance of this drug. So we are covering the part of the parasite concerning erythematolumefantrine. At the same time, we work in Mali with another drug, another one called piperquine. Strange names, I know. They're only familiar to me here. Piperquine has a characteristic, a very, very long half-life, some 20 days, 22 days. So it's quite nice to give as prophylaxis for protecting during say, the post-treatment period, which is important. The child is very fragile after the malaria disease, the episode. So it's good that it doesn't get malaria again. And that is great. The problem is that we also discovered that a new gene, well, we didn't discover, but we saw this gene associated with a collapse of the prophylaxis capacity of this drug. So, and this gene, this mutation, is spreading now in the places, making this drug much less effective. At the same time, we are involved with Novartis in the development of a new large, say, blockbuster, they like to call it, antimalarial, to replace the previous combination that involves lumefantrine. In fact, as you saw in the top, we found something that probably resists lumefantrine. Novartis doesn't know about this yet. In the last meeting, I decided not to speak too much about it. They will be very upset, but they will know soon when we'll publish. Now, the human side. The human side, Africa is a beauty and a nightmare. You know, if we think that we know all the diversity of pharmacogenes in the world, it's because we didn't go to Africa yet. And we knew a bit about that the African populations are the most diverse. So a couple of years ago, we made a collaboration in Gabon, in West Africa, in Lambarede, quite far away, 4 hours in a jeep, terrible thing, but a very, very nice place. And we just analyzed, it was a collaboration with the local authorities, and we analyzed 48 patients of malaria. And just as 48 patients that we analyzed only for 23 pharmacogenes, and we got a lot of new SNPs, a lot of new mutations, a lot of new stuff. Just 48 of that village. And in Gabon, there is at least 20 different ethnic groups with different languages. So they are really genetically separate. We don't know actually what is there. So this was a first glimpse of what we're facing in terms of understanding the diversity on the other side. But is this good for anything? Well, we don't know yet, but we know about one gene that Adrian knows very well too, CYP2C8, which is essentially the big factor metabolizing yet another antimalarial drug called amodiaquine. Amodiaquine is a very important drug. It's another big one. Amodiaquine itself has a very short half-life. Very short. It's a normal half-life, like the likes of 20 hours. But the metabolite, which is the clinically important one, stays there for 12 days. Again, very good for giving protection. In Mali, we made a very large study some years ago where we gave repeatedly treatments to people. People would come, get treated, we follow up, they get sick again, we treat again, we treat again, treat again. We did it for 2 years. They will never do it again. I will never do it again also. It was really a complex clinical trial. And we realized one thing when we start evaluating the levels of the metabolite. It's called acetylamodiaquine, by the way. It accumulates. So if you have a treatment in the next 30 days, until 30 days, it accumulates in the body. After that, continues to accumulate, but it's not really significant. But the first 30 days is really important. What we realize is that when we start genotyping people, is that accumulation is much bigger in the STAR2 patients. And this is not published yet. This is data from some weeks ago. Better off, the ones which are STAR2, they have more of these active metabolite, and they get more protected. Now, that's good news in principle. Because in another study in Zanzibar, on the other side of Africa, we realized that the same people that have this mutation, they get more protected. That's good, but they have more adverse events. So where do we go? That's something to evaluate. What is better, having more protection or having more adverse events? More protection is better, I can tell you. but it's something that we have to evaluate again, comparing this also with the reaction of the parasite. Finally, and to finish, because I wanted to be fast, we're starting to work with São Tomé and Príncipe. São Tomé and Príncipe is an amazing place. It's a small island nation, more or less in front of Gabon, but 200 kilometers inside the ocean. It is actually two islands, São Tomé and Príncipe. Now, what makes it special is that they are quite isolated. They had a lot of malaria, but they're trying to eliminate. And because of its isolation, small size, 200,000 inhabitants, they have chances to do it. Otherwise, it's very complicated to eliminate malaria. Further, the two islands, this map here, it doesn't really correspond to reality. Príncipe is way, way, way far away. It's like 180 or 200 kilometers. You have to go by plane there. And they are in two different stages of malaria pre-elimination. São Tomé still have a lot of cases, a lot, 1,000, 2,000 a year. Príncipe, much smaller, has only one or two. Now, they have the same drugs, but São Tomé is special. 200,000 people, everyone in the same island, but with a very complicated history. The Portuguese kept on putting slaves there. It was inhabited when it was discovered in bunches from different tribes, which meant that they segregated through history inside the island. There are 3 languages there. So there is probably genetically different populations that are pharmacogenetically different, all getting amlodipine, by the way, in big doses to try to put malaria down. And we have no idea, actually, if this has an influence on the efficacy of the drug. I can tell you one thing. They have a very curious way, an amazing way of controlling malaria there. When they see one case, they treat the case, and then they make a 28-day follow-up with weekly visits. That's a clinical trial. They just make informally clinical trial. And we're trying to weaponize it to make it a complete clinical trial that is really running all the time. When I saw, well, I've been in this business for a while, and when I saw the date of them, I realized that probably the treatment is not giving more than 75% efficacy, which seems a lot, but it's not a lot in malaria. Less than 90% in WHO wants to change a drug. So the idea is for São Tomé and Príncipe to be in the future, some years, we are working on it, to be the first place in Africa where precision medicine will be used to aid on the elimination of malaria. And that is not been thought until now, because until now, in most places where I talk, precision medicine is seen like not something for Africa, for God's sake, it's too expensive, it's too this. It's not true. It's just not true. It is possible to do things. And nowadays, the last 2 years, because of COVID-19, a lot of research and health centers in Africa received a lot of equipment, real-time PCRs, even next generation sequencing. They have sort of the capacity now to start thinking in these terms. And that's where I hope we're going from now on. And if anyone in Extremadura is interested in kind of Extremadura in the middle of the ocean, but we're only 1/5 of the population, it will be great to collaborate. Thank you very much.
Thanks a lot, José Pedro. I will introduce you to the group of microbiology here later on, probably Dr. Ciro Perez Giraldo, which is there. So, good friend.